Hello everyone, I'm Shweta doing second MSc microbiology. We are going to discuss about biofilm formation in environment. Introduction. Microorganisms naturally or artificially attach themselves to either biotic or abiotic surface, forming a complex matrix consist of polysaccharides, protein, and extracellular DNA known as biofilm formation. Biofilm provides microbes a unique environment that protects them from environmental hazards. Biofilms formed by single species is predominant in nature. However, polymicrobial biofilms formed by bacteria, algae, fungi, and protozoa also occurs in nature. In nature, environmental biofilms exist in lakes, rivers, and soil and rocks, aquatic plants, sediments, and in wetlands. Many microbial species are able to change their lifestyle like free living or attached depending on their physiological status and physiochemical conditions in their surroundings, taking advantage of the greater availability of organic matter in suspended particles and surface. Biofilm formation. A biofilm may also considered as hydrogel, which is a complex polymer that contains many times its dry weight in water. Biofilms can attach to a surface such as tooth or rock and may include a single species or a diverse group of microorganisms. Subpopulations of cells within the biofilm differentiate to perform various activities for motility, matrix production, and sporulation, supporting the overall success of the biofilm. The biofilm bacteria can share nutrients and are sheltered from harmful factors in the environment like desiccation, antibiotics, and their host body's immune system. Moreover, the different microbial species in consortia of biofilms, each with different metabolic degradation capable, could degrade several pollutants either individually or collectively. A biofilm usually begins to form when a free swimming planktonic bacterium attaches to a surface. Origin of biofilms. Biofilms are thought to have arisen during primitive Earth as a defense mechanism of prokaryotes, as the conditions at that time were too harsh for their survival. They can be found very early in Earth's fossil records, about 3.25 billion years ago, as both bacteria and archaea, and commonly protect prokaryotic cells by providing them with homeostatics, encouraging the development of complex interactions between the cells in the biofilm. Types of biofilms. Biofilms can contain many different types of microorganisms like bacteria, fungi, algae, protozoa, and archaea. Each group performs specialized metabolic functions. However, some organisms will form single species films under certain conditions. First, the microbial biofilms. It is capable of acting as a biomonitors, which allows the detection of emerging pollutants in addition to their high tolerance capacity to chemical substance or other pollutants. That results in the acceptance of almost any environmental stress that occurs. For example, large areas of land dedicated to agriculture, aquatic environments, and food chain are exposed to high use of pesticides, which cause negative effects on the abundance and diversity of species. On the other hand, biofilms allow the absorption, mobilization, or degradation of environmental pollutants. In certain places near contaminated areas, there are bacterial biofilms ready to persist, survive, and better manage the adverse environment. The second one is algal biofilms. It can be established on any surface that receives sufficient light and moisture. These films are complex communities of both autotrophic and heterotrophic organisms. These films can contain different microorganisms from diatoms and cyanobacteria or protozoa. Microalgae biofilms composed of cyanobacteria and or green microalgae are ubiquitous, distributed in almost all the photoaquatic environments. Microalgae can be used in aqueous treatment process, secondary and tertiary treatment for a range of purposes, some of which are the removal of coliform bacteria, reduction in both chemical and biochemical oxygen demand, removal of phosphorus and or nitrogen, as well as the removal of heavy metals, since they can be further converting them to more amendable forms. Stages of biofilm formation. It takes place over five main stages, including: first one is initial reversible attachment, second, the irreversible attachment, third, maturation stage 1, fourth, maturation stage 2, and the last, dispersion method. The first one is initial reversible attachment. Bacteria generally attach to a surface that is rich in organic molecules, example, nutrients, salivary proteins, and large macromolecules. These molecules promote the attachment of bacteria to the surface. Initial attachment is mediated through weak van der Waals force, which later turns to stronger dipole-dipole interaction, hydrogen ionic or hydrophobic interactions. There is a strong adhesion receptor mediated attachment. The second one is irreversible attachment. Initial reversible attachment further changes to the irreversible attachment. In this stage, the force of attraction are greater than the force of repulsion. Initially immobilized bacterial cells attach to the surface irreversibly. The structures present on the surface overcome the physical repulsive force of the electrical double layer of the cell and consolidate the interaction between bacteria and the surface. The hydrophobic interactions between the surface and bacteria also reduce the repulsive force between them. The third one is maturation stage 1. The bacterial cells start communicating in this stage by the production of AA signals, which results in the expression of biofilm specific genes. The bacteria start producing extra polysaccharide, which stabilizes the biofilm. In this stage, the thickness of biofilm increases up to 10 μm. The fourth one is maturation stage 2. In this stage, the thickness of biofilm further increases to 100 μm. Multi-species microconsortia develops on the surface, which results in increase in substrate exchange between bacteria, distribution of metabolic products, and removal of toxic end products produced by the bacteria. In this stage, biofilm adapts with the external conditions by manipulating its structure, physiology, and metabolism. The fifth one is dispersion. Dispersion of bacteria takes place and bacteria return to motile form. In this stage, the microbial community produces different saccharolytic enzymes which break the biofilm stabilizing polysaccharides that releases the bacteria present on the top of the biofilm and colonizes to the new surface. The microorganism upregulates the expression of flagella proteins and bacteria return to motile form to translocate to a new site. Environmental conditions of biofilm formation. It can influence both bacterial properties, which is mediated by changes in gene regulation, and or cell surface physiochemical properties, and surface proteins, which is mainly through physiochemical changes. They can also control concentration of the second messenger CDIGMP, which regulates biofilm related factors such as cell appendages, surface proteins, extrapolysaccharides, and cell motility. The environmental pH can have a major effect on the formation of biofilms. It can attach to the surface, which is the first step in biofilm formation, has been shown to influence by this factor. The bacterial cells within the biofilm can withstand pH changes compared to the free floating cells. For example, under highly acidic condition, the bacterial biofilms gel-like structures can help in reducing the rapid diffusion of ions and allow for the development of a pH gradient within the extracellular matrix. Biofilm control. Controlling biofilms can be challenging due to their complex nature and resistance to traditional cleaning methods. Here are several strategies commonly used to control biofilms. First, physical removal. Physical removal. The biofilm can be physically removed through scrubbing, brushing, or high pressure washing. This disrupts their structure and reduces their viability. Second, chemical agents. Various chemicals can be used to disrupt biofilms. The third, antimicrobial agents. Kill or inhibit the growth of bacteria in biofilms. The fourth one is enzymes. Degrade the extracellular matrix of biofilms, making them easier to remove. The surfactants. disrupt the biofilm structure by reducing surface tension and breaking up the matrix. Disinfectants kill microorganisms in the biofilm and prevent the regrowth. Biocides. These are chemicals that can kill microorganisms within biofilm. They are often used in combination with other methods to enhance effectiveness. Biofilm resistant materials. Using materials that are less prone to biofilms formation, such as certain plastics or coatings with antimicrobial properties, can reduce the biofilm buildup. Applications. Biofilms have various industrial and environmental applications due to their unique properties and capabilities. Here are some notable applications. The industrial use: wastewater treatment, bioremediation, biocatalysis and biosensors, corrosion control, biofilm based materials. The environmental use: maintaining the soil health, aquatic ecosystem, biofouling management, biogeotechnical applications, and finally, biofilm based sensors for environmental monitoring. Thank you for your patience.